Welcome to the course Basic Electrical and Electronic Engineering. I am Indu Bhardwaj. In this video, we will discuss biosensors for environment monitoring. The material which is being presented in this video has been taken from internet site and ebooks, and this can be used for only academic purposes. So, biosensors are being rapidly used in our environment monitoring as your environment is being polluted by various harmful pollutants. And these harmful pollutants are being increasing day by day as the pollution is increasing. So we can use the biosensors for measuring the type as well as the concentration of the contaminants which are present in our environment. Generally, the primary measurement for this pollutants are we can use it for the monitoring of water, soil, and air pollutants. But in the long run, biosensors can also be used for the environment polluting alarm system for specific allergy, including your heavy metals, herbicides, pesticides, phenol compounds, etc. For example, your biosensors, which are you can say which are using antibodies as the recognizing elements, which are they're using immunosensors, aptasensors, they're genosensors, enzymatic biosensors. They are rapidly being used for monitoring and detection of various environment pollutants like your antibodies, aptameters, nucleic acid, enzymes, etc. respectively. So what are the benefits of these biosensors? Those which are normal sensors. So with respect to that, your biosensors are having some advantages like they have the ability for the portability. They can be brought to any of the places and they're quite small in size. So miniaturization is also an advantage. It is the measurement of pollutant with minimal samples. We need only some of the samples for finding out the results for the presence of the pollutants in the air, water, soil. So the commonly used biosensors for the environment monitoring are your optical and electrochemical sensors. So these we have already discussed in our previous videos when we were doing types of biosensors. Then we have covered this part. So we'll see how biosensors are being used in environmental. So biosensors are used in detecting environmental pollution and monitoring of mines, industries, and toxic gases. They're also used in the BOD measurement during the wastewater treatment. It's also used in the detection of pesticides in your river water contaminations. When the river water is contaminated with high metal ions, then we can use biosensors for their detections. It is used for detection of your polychromatic hydrocarbons which are present in the air and the determination of your organophosphates. So all these are the applications of biosensors for determination of environmental pollutants. So we'll discuss some of the biosensors. Like one is the gas biosensors. Gas biosensors are used for detection of your sulfur dioxide, methane, carbon dioxide. Besides that, we have microbial biosensors. It is some more specific. Next one is your immunoassay biosensors. It is using trypsins, carbamates. These are being on this. Then we have BOD sensors. It detects the level of organic pollutions. So this requires 5 days of incubation, but our BOD sensor using yeast with oxygen probe, it can take only 15 minutes to detect your organic pollution. Then we have some more biosensors like your miscellaneous biosensors. So graphite electrode with cyanobacterium and your Cyanococcus. With these electrodes, your electron transport inhibition during your photosynthesis due to the certain pollutants like herbicides. So presence of these can be detected using with the help of these biosensors. Besides that, we have biosensors having their PCBs, then that is your polychlorinated biphenyls and chlorinated hydrocarbons and some other organic compound can be detected with the help of these. So this way we can use the biosensors in various applications like for the detection of organophosphorus compounds in the water. So we will discuss the pollutants one by one. Then similarly we have toxicity assays, which are based on the use of luminescent bacteria. So these bacterial bioluminescence has proved to be the convenient measure of your cellular metabolism, which is a reliable sensor. Similarly, your cell sense is also an amphiometric sensors. Amphiometric sensors we have already discussed. So it is a type of amphiometric sensors that incorporates your Escherichia coli bacteria cells for rapid ecotoxicity analysis. Besides, cell sense is one of the newly rapid toxicity assessment. It is one of the best methods, and it investigates the toxicity of 3,5-dichlorophenol and other phenols in the wastewater for the determination of non-ionic surfactant and the benzene sulfonate compounds for the analysis of wastewater treatment. So we will discuss the pollutants one by one. First one is the heavy metals. So for determination of these heavy metals like your copper, cadmium, mercury, zinc in the environment, it is important to determine them because of their high toxicity. They are very toxic metals. So their level is continuously increasing in our environment. So it is harmful for the marine organisms. So for sensing these, we use we use the luminescence based bacterial sensor strains which detects these heavy metals. Next one is our polychlorinated biphenyls. So these are the also very harmful pollutants for the environment, and they are used as industrial chemicals, particularly in dielectric fluids in the electrical transformers and capacitors. The high toxicity of these PCB contents represent a risk to the public health. They are that's why they are quite harmful. So different biosensors are used for their determination. So PCB in the environment, and these include the DNA biosensors with chronopotentiometers detection and various immunosensors with the fluorescence. So they can be used for these PCBs detection. Next one is our dioxins. So dioxins are released as a byproduct in various chemical processes. So biosensors which are used for detection of these dioxins, these are the SRB biosensor, and they are developed by Shimomura for the determination of PCB. They have employed in the determination of dioxin 2378 TCDD, and similarly some other biosensors for the detection of dioxin like chemicals based on the recombinant mouse hepatome cell. These are also being developed. So we have different sensors for the detection of the presence of dioxins. These conventional dioxin analysis requires laborious multi-steps cleanup process that increase the cost of each analysis. So a significant number of immunoassays for dioxin has been developed in an effort to provide simplified routine analysis. So for this we are using different sensors. Next one is the pesticides. So pesticides we all know, and the detection of these pesticides we are using some different biosensors that are based on photosystems. So this photosystem second is being used for this. So they are able to detect herbicide in the environment. Almost 30% of the herbicide, which include your phenyl, urea, triazine, phenolic herbicide, inhibit photosynthesis electron flow by blocking PS-spin quinine binding site and thus modify your chloryl fluorescence. Next one is our antibiotics. So these increasing use of antibiotics for the therapeutic purpose or as a growth promoter in our dairy cattle and as a feed activity in the fish farms, it is used. So for detection of these antibiotics, our commercial BioCare 3000 sensors are being used, and the cross reactivity between the two sulfamides and the sulfamethoxine and the furosemide. So sulfonamide sometimes causes allergic reaction, whereas the effect in the human immune system is of high interest for their therapeutic applications. And different sensors are, besides this, some more sensors are being developed by some authors, like you can say not some authors, some researchers like Hansen and Sorensen. They have presented some more sensors for detection of their antibiotic. This pollutant is your phenol. So phenol component originate from the paper or the pulp industries, or from the production of your drugs, dyes, or antioxidants. So these phenol compounds, especially your chlorophenols, these are quite important because of their high toxicity. So they need to be detected. So your Perleda scientists, they have developed an amplitude biosensors with thyrosinase, that is a polyphenol oxidase with the relativity wide selectivity for the phenol compounds. So they have developed the amplitude biosensors which can measure the presence of phenol in the environment. For chlorophenols also we have developed the biosensors. So these biosensors are your lux-based biosensors. They are used to assess the toxicity of the paper mills, which are being using some metals like cadmium, copper, and your PCP. Next one is your toxins. So toxins are also very harmful. They are the heterogeneous group which is capable of affecting different biochemical processes, including our membrane function, ion transport, transmitter release, and DNA and protein synthesis. So for detection of this, the integrated optical sensor is being used. Besides this, the impedance-based immunosensor is also being prepared, and it is prepared by an ultra-thin platinum film with an immobilized layer of antibodies against the streptococcal enterotoxin. So these we can detect with the help of these immunosensors. Next one is our inorganic phosphates. They are also very helpful. They are found in the surface water and is used as a measure of the degree of eutrophism. And the sensors which detect is we are using the phosphate sensor, or you can say enzymatic phosphate sensor for the detection of the phosphate compound in the inorganic phosphate. So next one is our BOD. BOD is biochemical oxygen demand. So BOD sensors have been developed in the market by various manufacturers in both biofilms and bioreactor type configurations. So most of these commercially available biosensors are flow type systems that can be easily automated, but they generally require high maintenance to prevent fouling and clogging. So many BOD biosensors are used for the determination of high BOD values as the industrial wastewater, and they are not to adopt in the measurement of low BOD values. So for detection of these, we use the optical fiber biosensors for evaluation of low BOD values in our river waters. Next one is the nitrate. The increasing nitrate level are being found in the ground and the surface water. It is of great concern because they harm the water environment. So biosensors containing immobilized denitrifying content, they can be used for the determination of your biosensor, this nitrate compound. So microscale biosensors are used for this nitrate determination, and they are used for in-site monitoring in an activated sludge plant. So biosensor was based on the diffusion of nitrate through the tip membrane into the dense mass of the bacteria, converting the ions into the nitrous oxide with the subsequent electrochemical detection. Next pollutant is our hormones. So endogenous hormones of humans as well as the animals origin have been reaching the environment for thousands of years. Besides endogenous hormones, exogenous steroids are used for the growth promoters in the several countries, have become a matter of great concern. So for them also, we have the biosensors. Although low concentration of hormones such as your estradiol, estron, they have been found in the water. Their widespread use of their capability to introduce response in fish at the concentrations as low as Ng have altered the scientists to potential dangerous consequences of their presence in the aquatic environment. So these hormones are affecting the aquatic environment also. So for them, we use optical immunosensors in the real water samples for detection of these hormones. Next one is our endocrine disruptive chemicals. So these chemicals are also affecting the environment and they are contaminating the environment. So for this, we are using SRP biosensors, BioCare. They have been used in the determination of estrogen and xenoestrogens. Next one is our surfactants. So detergent product use surfactant as the basic active components. So for the determination of these surfactant, your amphiometric biosensors are used for the detection of anion and surfactants, which was constructed with pseudonomous rethions. Besides that, your microbial biosensors enable the detection of surfactant with the high selectivity and sensitivity and reproducibility. Next one is our green fluorescent based pseudonym fluorescence strain biosensor. They are also used for the same. So different scientists have proposed different biosensors for the determination of your surfactant. Amphiometric biosensors for naphthalene found in the contaminated soils. Besides that, we have pH or your carcinogenin compounds, which are generally found in the incomplete combustion. So for them, amphiometric biosensors are being used. Next one is our bioremediations. So it is an application of microbial capacity to transform complex organic molecules into the simpler organic molecules. So these biosensors can monitor these parameters with the help of better control the biomedication process. The molecular biosensors are used for the determinant of this. Next one is microorganisms. So like your bacteria, viruses, other microorganisms, these are popularly polluting the untreated and tasted water, which implies a worldwide public health problem. So for them, we are using the sensors at the concentrations down to the 106 cells per milliliter. Recently, a number of piezometric biosensors are being used for this. So et al. Described biosensors for determination of E. coli in your water samples by immunochemical potentiometric altering biosensors. Besides that, Haseba et al. Describes the amphiometric biosensors for detection of E. coli in the wastewater. So these are some commercially available biosensors for the detection of environment pollutants. Like these are the instruments like your BioCare, Remedios, then BAS BODs, and these are the biological elements which are being used in BioCare instrument. We are using biomolecular interaction as a biological element, and the transaction we are using as optical, and the analyte is the sulfonamide pathogens. So this is the complete table, which is these are the biosensors which are commercially available in our market for the detection of environment pollutions. So measurement of ecological climate changes under the pins for the formula of effective management strategies for sustainable use of and the protection of marine environment, and the sensors are traditionally used in the marine studies to determine the physical parameters, but there is increasing demand for the real-time information about the chemical and biological parameters, and these parameters are currently measured in the sample collected at the sea and subsequently analyzed in the laboratories. So we have different sensors for measurement of toxins, or you can say the pollutants under the seas, under the in the rivers also. So we can save the marine life also. So if you have any doubt in this topic, so let me know. Thank you.